On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Welcome to the Life Science Success Podcast. My name is Don, and I'm the host. I am a consultant in life sciences, and I help companies manage complexity and increase performance. Today on the podcast, I am joined by Akunna Iheanacho. Hi, Akunna.
Hello. How are you doing, Don?
Very good. Yeah, thanks so much for joining me. Can you tell the listeners just a little bit about yourself?
Sure, I'm happy to do it. So I'm the child of immigrants. My father, Morris Iheanacho, is from Nigeria, and my mother, Shirley Iheanacho, is from Barbados, and I'm very proud of my rich heritage and culture. And I grew up in Huntsville, Alabama, in a very small, close-knit community with my two older sisters, Ngozi and Chioma. And from a very young age, my parents instilled within us the importance of giving back and having a positive impact on our community and on the world at large. Presently, I am the Chief Scientific Officer at Texcell North America, where I provide scientific and technical direction for all programs and business units. And I'm a subject matter expert with broad biotechnology experience and expertise in areas that include process development for large-scale current GMP manufacture of vaccines, AAV gene therapies, and other biopharmaceutical products. So along my journey, there have been countless different individuals that have poured into me and patiently shared insights that I would continue to mull over for many years. And some of these individuals introduced me to the wonderful and fascinating world of science that still captivates me even now. This is also one of the reasons why I am so passionate about STEM outreach. I remember how those individuals really opened up new worlds for me, and now I have a chance to pay it forward. So I also serve as the vice chair of the National Committee of Young Women in Bio, where I have the opportunity to interact with the next generation of talent, and I have a chance to help them effect positive change through science, technology, engineering, and math. I'm also a very active member of Women in Bio, which is an organization that promotes diversity and inclusion across the life science sector.
Yeah, it's interesting, the women in bio tie for sure. I mean, I've spoken to a few people. We actually have a few guests that are coming up as well that are in women in bio. And I guess, tell me a little bit more about your involvement with STEM. I mean, so when you were going through school, it sounds to me like you somehow, did something with STEM. Can you tell me a little bit
Yes,
more?
I did a few different things with STEM. So I guess how I started to break into the field of life sciences and get involved with STEM, I was trying to satisfy my curiosity. I liked asking questions and I liked taking things apart and trying to put them back together. And so decades later, I believe that this is still my approach. I like to sort of peel back the layers and try to figure out what's going on under the hood. So, as an undergraduate, really it began before being an undergraduate. I had an opportunity to participate in a lot of different summer enrichment activities from middle school through high school, and then even through college. Once I was in college, there were a number of internships that I was able to participate in. And one in particular really had a tremendous impact. It left an indelible impression on me. So there was a summer research institute. It was called the UNCF Summer Pre-Medical Institute. And it was through a collaboration or partnership between Fisk University and Vanderbilt University Medical Center. And so during this internship, I had a chance to participate in taking some courses, research symposiums. I had an opportunity to shadow physicians in a very clinical setting. And then I also had a chance to work in a type 2 diabetes mellitus clinic. And what really intrigued me about the research that they were doing, there was this one endocrinologist in particular that sort of took me under their wing. And it was almost as if he made it his mission to see, let's see if I can really get this young woman to think critically about what we're doing. So he would bring journal articles and discuss them. He would relate things back to items that we had discussed or covered during clinical rounds. And so as a result of that experience, he really challenged me to gain a better understanding of type 2 diabetes mellitus. And he said that as I accompanied him in the clinic, if I really had an understanding of this disease, since it impacts most organ systems in the body, if I could grasp its particular pathology, then that would unlock doors in my understanding, not only of organ systems, but of physiology as a whole. And so this idea really persisted with me, and I would later revisit it after college. So I went on to pursue my master's degree in chemistry. And while there at the University of Virginia, I utilized many different in vitro approaches to evaluate this group of small molecules. So I was in a medicinal chemistry group. And so the synthetic chemists would produce these, what we hoped would be novel chemotherapeutic agents. And then it was my responsibility to figure out what is their mechanism of action? How are they able to actually kill these cancer cells? In the hopes that we would be able to produce some novel chemotherapeutic agents. And so from there, I still wasn't done. I said, there's some more questions I could maybe answer, some more things that I could do. So for a time, I worked at Johns Hopkins University School of Medicine as a senior research technician. And then while I was there, I decided, okay, I want to continue my education and I'm going to pursue doctoral studies. So I continued there at Johns Hopkins University School of Medicine, and I pursued my doctorate in cellular and molecular physiology. And my studies really focused on the structural and functional characterization of a group of proteins that were involved in different cell signaling pathways. So those that were dysregulated in type 2 diabetes mellitus, as well as those that were dysregulated in cancer. And I was excited to learn some of these different approaches and expand my toolkit or my arsenal of investigative techniques. So part of that, I learned how to use structural biology and mouse models in this multidisciplinary approach to perform the functional and structural characterization of a group of proteins. And I was excited because now I was not only able to consider how these organ systems interacted at a macro level, but I was now also able to evaluate some of these interactions now at the molecular level. So after I left Hopkins, I went on to pursue a career in industry because I really wanted to translate my knowledge and some of my expertise into developing therapeutic products. So I went on to work at a contract development and manufacturing organization. At the time, it was called Paragon Bioservices Incorporated. It is now called Catalent Cell and Gene Therapy.
Oh,
And
sure.
while I was there, are you
Yeah.
familiar with that?
Yeah, I
Okay.
know Catalent. Sure.
Yes. So while I was there, part of my responsibilities included developing CGMP processes for biologics. So my work focused specifically on downstream process development, formulation, and technology transfer of therapeutic proteins, monoclonal antibodies, vaccines, as well as AAV gene therapies.
I mean, you have had quite the career, and it seems like you're still really early on in your career. So, I mean, really quite the journey overall. And now you're at Texcell, and you're the
Yes,
chief scientific officer there. Can you tell
yes.
everybody a little bit about Texcell and what it is that they're trying to solve?
Certainly. So Texel is a global organization. They have sites in Frederick, Maryland, which is where I work. We also have organizations in Taiwan, Japan, France, and China. And at Texel, we like to assist biopharmaceutical manufacturers or organizations or institutions by offering contract testing services that include viral clearance evaluations, viral safety testing, and cell banking. Essentially, we offer these services, this portfolio of services, so that we can ensure or help to ensure the product safety, whether they're using particular cell lines that are going to be used as the hosts for these different therapeutic products, or whether it's going to be a potential therapeutic and we want to evaluate the safety profile of it to ensure that once it's going into human beings, it will be safe and efficacious. So, our goal really there at Texcell is to alleviate some of the stress that is associated with designing and executing some of these viral clearance studies in support of regulatory submissions. Often that's a pain point for some organizations. So, from early investigative studies to late stage support of biologics license applications, we offer many different options and we have a very dedicated team ready to partner with these organizations.
Yeah, I mean, it's one of the things I know that doesn't come top of mind to me, in terms of the things that happen overall in terms of drug development. But absolutely, it's critical, especially before, submission dates and things like that to complete these studies as well. So,
Yes.
yes, absolutely critical. So, in terms of your career and some of the things that you've done, I'm just curious to find out what is one important lesson in your career that you found that really shapes what it is that you do.
So, one very important, if I can only choose one, because I always feel like there's three or four or five, right? But if I could only choose one, I would say that it's embrace failure. Don't run from it. So, I believe it was David Duchemin who said, failure is a much more faithful teacher than immediate success. So, embrace those challenges and view setbacks as opportunities to utilize your critical thinking skills. Carve out some time to actually learn from past failures. Truly being innovative can be risky at times, but some of my most rewarding moments have come from pausing to gain a greater understanding of what did not work, and then using that experience or those lessons learned to inform either the next phase for that particular project or for other projects that came later. So, past failures truly have the potential to inform future work. And I think that true knowledge to me is more than just the mere acquisition of facts, but it's also the ability to think critically and analytically through observation and problem solving.
I mean, it is such an important lesson, and one of the things that not a lot of people want to be very open about, in terms of, hey, I really fell down here, and this is how, I've maybe learned that lesson either. And
Right.
I am thankful for some of the people that I know, at least, that were willing to kind of share, items like that in my career
Mhm.
history, because you do learn so much more from not just your own failures, but from other people's failures as well.
True. That is so true. That is true. Yeah. I think that's
Go
something
ahead.
that we should say more often. Um, that, hey, if it was easy, someone else would have already done it.
Yeah.
So if you want to be innovative, you have to embrace failure.
Yeah. And I mean, if it feels so much like a part of scientific discovery is that, right? That
Mhm.
you sort of, maybe you didn't exactly solve the problem the way that you thought that you would, right?
Yes.
Maybe you started out with this grand vision that, hey, look, by the end, I've solved this particular problem. However, the things that you did learn and the things that you sort of been able to eliminate along the way will help the next researcher just as much. And I know
Definitely.
even in my own dissertation, I was surprised to find a few years after finishing my dissertation, people had sort of referenced my work as well, and just knowing that somebody knew both what went well as the things that I learned that didn't go so well in my dissertation. It was helpful, I at least for me to see that it was being carried forward, that other people were
Right.
referencing the work.
Definitely. Definitely.
Yeah. So, what is any advice that you would give somebody that's just getting started in life science? It seems like there's so much going on, and such an opportunity to be able to guide somebody that's coming, brand new into life science as well.
Sure. Determine your desired end. So if you begin with the end in mind, it will help you frame what your next step should be. So some questions you could ask yourself: what is the desired outcome? And then start to consider, what will it take to reach that target or to reach that goal, that end, that outcome. For example, let's say that you're going to draft your manuscript. You've completed your experiments, you're a grad student, and you're working towards either your master's or your, let's say, your doctorate, and you're trying to figure out, okay, how am I going to start to put together my publication? And one thing I would say is, okay, as you're reviewing your data, think about what story it is that you want to tell about this story. Because I really think that the data should be that. It should be a story where people can see a clear progression from start to finish of maybe what your hypothesis was and then what your final conclusions were. So you can start by, or one trick that helped me, I would start by creating the figures and the figure legends first. So after I compiled all the data, I would see, okay, so what figures can I make based on this data? So I actually started, some people would say backwards, but for me, I found that it was a better technique for me. It helped focused me in. So I would draft those, create those figures, create the figure legends. Then I would write my conclusions and then go back and draft the other pieces.
Mm.
So I just feel like it's a good illustration of determining your desired end and then doing what you need to do to achieve it. So that's one thing. Another thing that I would tell someone just starting out is really begin now to understand the importance of networking. Look for opportunities to grow your professional network. So, at research symposiums or poster sessions at conferences or annual society meetings, take any and every opportunity to interact with others. These are people that, if they're not your colleague now, they might be your colleague in the future. Interact with them, engage them in conversation. You're there at the symposium or you're there at this conference, so you already have something in common with them. Use that as a framework to sort of stretch out of your comfort zone and start to learn about what others are doing in the field and start to build up your network. Actively seek out mentors and advocates. You'll need both throughout your career. Not only the mentors, but you'll also need advocates, especially as you're starting out. So, Women in Bio and the American Chemical Society have both given me outstanding mentoring resources. My membership in Women in Bio has also greatly strengthened and expanded my professional network. Also, think about the colleagues or the coworkers that you're interacting with now on a daily basis. Maybe those folks might serve as individuals that you collaborate with either later on in different projects or future ventures. So keep those lines of communication open. So then I guess, yes, the two main things I would say: determine your desired and understand the importance of networking.
Yeah, for as for number two, I mean, I would say I have two items that I would add to that. I mean, one, um, I was with GE Healthcare for 17 years in my career, and along the way, I met a lot of people, and along the way, also LinkedIn was started, and so. I remember I have this thing LinkedIn and I have no links. And then, I started adding, essentially people in my LinkedIn network, here and there. And I think as I got ready to leave GE, I had something like 1,000 connections,
Oh,
people that
wow.
I had met along the way, either in GE or at conferences or whatever, but at least they were connections. And then I remember talking to somebody shortly after I left, and he said, well, I didn't do any of that. I have about 100 connections on LinkedIn. And I felt like I really had, a lot of connections now since leaving. I've met people that have maxed out their LinkedIn connections, which is something like 30,000 people. So I don't know that many people yet. But it's well worthwhile to have, some form of a connection with people. And then the other thing is, I know there are people that I mentor as well that,
Mhm.
in our discussions, they just say, well, I don't know how to do this. I don't know how to get started at conferences. And my sort of point of reference, and it kind of goes to something else that I've used in life, which is, how do you get to know somebody? I mean, typically one, where do you live? What's the weather? There are places that we can all start, and then all of a
Yes.
sudden a conversation starts, and then next thing you know, you have somebody that at least something more about. Now, if you really can't do it, the one last suggestion that I like to give people is, if you've ever seen the matrix for a bingo card, draw yourself up a little list of, like bingo squares where you can ask people at conferences. So, what is your favorite color? Ask people, unusual things. Do you live in a 1-story house or a 2-story house? I don't know. I mean, maybe they're just things that you could really sort of base your conversation on. I study this, and, you studied, it sounds to me like diabetes and cancer in school. So, I mean, there are places where we can all start, with
Yes.
that conversation. It's getting it started. That's the hard part. Afterwards, it's a lot easier.
Yes. Yes. Those initial steps are the hardest, I think. But after you overcome that, then it's a lot easier.
Yeah, for sure. So what are some of the biggest challenges that you faced this year?
So I would say in my field, really now, gene and cell therapy, gene and cell therapies are on the rise. So there are several of them that have won approval, as well as other advanced medicinal therapeutic products. And so now we're starting to see these treatments of rare diseases that at some point were thought to be untreatable or incurable. And now they're finding treatments for them. So in in evaluating or considering some of these new novel therapies that are coming to the forefront now, we are discovering that we don't always know what we don't know. And that in and of itself can prove challenging. Some questions might be, so what is the best way to develop a manufacturing process for a gene therapy when you have limited knowledge and you have short clinical timelines? Is there a good sense of the viral clearance that could be obtained for each process step or each downstream process step? What do you do when the technologies that you would typically use for virus removal or for virus inactivation are incompatible with the manufacturing process itself, or the manufacturing process that's used for these complex biologics? What sort of regulatory barriers might you encounter? And then how you distinguish viral contamination from a viral vector or from viral vectors, plural. If virus filtration is not an option, what other ways have you identified or can you identify as a potential way to remove viral contamination? How do you keep this product intact
Mm.
uh, while trying to inactivate viral contaminants? So there's a whole host of challenges and approaches that we need to consider specifically for these complex advanced medicinal therapeutic products. And some of these challenges that we're considering at Texcell, we really try to customize the approach. So with every partnership or with every client, we really look at their process and how what their risk assessment is, and we factor all of that in when we're designing or coming up with the study design for the viral clearance studies. Um, so yeah, I would say that those have been some of the biggest challenges that we face this year. Trying to think of new ways to approach some of these more complex, because the problems that you would face when you're evaluating a monoclonal antibody are very different from the challenges you might face when you're evaluating a gene therapy.
Ah, yep. Completely understood. And then, what's the one thing that you're going to start doing this year?
One thing I would like to do this year, I would like to learn another language. I haven't
Ah,
settled yet on which one, but I would like to at least begin the process of learning another language.
that can be it could be a fun adventure. I've tried a few times. Not it, it I'm not that successful and not brave enough yet with any language to actually step out in front of somebody that speaks it natively and try it either. So that's a whole nother I guess hurdle that you have to get over and you know, really be open to, you know, sort of speaking the language in front of, you know, people that know it well enough, and then, you know, it
Right.
seems to help as well.
Right. I think that's the truest test, right? Have you
Right.
have you really have you really learned it?
And what's the one thing you would like to stop this year?
One thing I'd like to stop this year is bringing work home. I want to really, once I have left work, leave it there. Because I find that when I do that, I'm so much more fresh, and it's almost as if my perspective, I give my mind time to stop thinking about it for a while, so that when I return to it, I often will have a fresh perspective or a different outlook. So I'm really trying to do a better job of leaving work at work.
Yeah, I've found that there's so many people, especially, you know, kind of where we are right now with COVID, that have just picked up even more work than they had well in advance of, you know, the time that, you know, we've had COVID in the world. And
Yes.
with that, I mean, we just don't get a break, and it just seems like we can, you know, work nonstop and things like that. I know one of my personal things is just having time in the morning, not necessarily you know, in true meditation or anything like that, but just taking that mental break where I'm not thinking about emails, I'm not thinking about anything else. I'm just kind of, you know, more or less trying to take that mental time to
Mhm.
get a bit of break before I start my day each
Mhm.
morning. And I have a good friend that's leading a company where they're the one thing that they're doing right now is that they're granting people the first hour of the day. They're paying them for this first hour, but it's their hour, whatever they want to do. They're not to do work things during that hour, but whatever they'd like to do during that time is up to them. And
Oh, that's
it's just
great.
an interesting way to kind of, you know, draw people in, but also, you know, get people to kind of use one of the better hours in the day for themselves as well.
That's great. I wonder if it sparked more innovation throughout that organization.
Yeah, I think we'll have to see what, you know, what happens in the long run with
Right.
the output, you know, with it. But yeah, I mean, I think there's a lot of great things that could come from it. To me, it kind of reminds me of the you know, the Google 80-20%, you know, kind of split where you can go work on things that you want to work on, but you know, it's a little bit of a different twist to that same idea.
Yes.
So there are three questions that I like to ask every guest. What inspires you?
So, being out in nature as well as observing art in all of its different forms. Both of those activities really, really inspiring.
And you're in Maryland, so there's a lot of great things that are happening right now in nature. I mean, I would imagine, are the cherry blossoms all gone now, or are they just happening now? Do you know?
So not all gone. Some of them, depending on where, some have. We did have some storms that kind of knocked a lot of blossoms off, but I still see some on some trees. So they're still in bloom. They're not
Yeah.
completely gone yet.
And a lot of, I mean, just a tremendous amount of great art in Maryland as well, and good great place for science and innovation too.
Yes.
What concerns you?
What concerns me most right now is the vast amount of disinformation and misinformation surrounding or regarding COVID-19, vaccine development, and quite honestly, the misinformation and disinformation surrounding life science or biomedical topics in general.
Hmm.
And it's really made me consider, what can I do to combat some of these myths or combat some of this misinformation? So I've really tried to give seminars, give webinars, discussing some of these topics and trying to explain them, hopefully in terms that anyone could understand, so that they have a better understanding of not only how different therapeutics are approved or win approval before they will go into a human being, but also to sort of share with them, I guess, some of the first principles around science so that it's not so mystifying and so scary, maybe.
Yeah, I mean, it is very important now. And I mean, just in general, being able to understand, what you should really be afraid of or what you shouldn't really be afraid of would be a nice place, I think, for a lot of people that are not in this field to understand. And so,
Mhm.
I mean, it's definitely sounds like some important work. And what excites you?
What excites me? So, doing work that positively impacts others. So, it's both thrilling and rewarding to be an ambassador for science, whether that's mentoring young people interested in STEM or supporting IND enabling or BLA enabling programs for gene therapies or monoclonal antibodies or other medical devices, or facilitating the development of diagnostic tools. So it's tremendously thrilling to assist in bridging the gap between scientific discovery and then the delivery of some of these new therapeutic products to market. In fact, one of the most rewarding moments of my career was when Zolgensma, a gene therapy treatment for children diagnosed with spinal muscular atrophy, received FDA approval.
That is amazing. And I would imagine, I mean, I guess correct me if I'm wrong, but I would imagine your time frame between the time that you see something um, to the time that it goes to market is probably a lot shorter overall. Because I would envision that your viral load studies and things like that are fairly late in the process, correct?
Yes, yes, they're fairly late in the process. I guess in the case of Zolgensma, though, I actually helped develop the manufacturing process and validated this process for it. So in this instance, I had a chance to help steer and guide and validate this process that developed this therapeutic that then went on to clinical trials and, yes, wound up being an improved product.
Very good. Well, Akunna Iheanacho, thank you so much for being on the Life Science Success Podcast today. I greatly appreciated our time and our conversation together, and just wish you all the best of luck in your career.
Thank you so much. It was a pleasure speaking with you. Thank you for the invitation.
Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it, and last but not least, rate the podcast. Thank you again.